This is Agriculture Today, and I'm Shelby Varner with the K-State Radio Network. The show starts with Laura Miller, K-State Associate Professor of Veterinary Virology, as she explains research of using mealworms to help protect poultry from highly pathogenic avian influenza. Laura continues today's show as she discusses how this can be used for wild birds and other species, as well as highlights the teamwork on this project. K-State dairy specialist Mike Brouk ends the show as he passes along data from 2025 that shows an increase of 45,000 dairy cows in Kansas, which helped produce a banner year for milk production and is expected to continue in 2026. That and more is coming up ahead. You're tuned in to Agriculture Today, and we start our show discussing how highly pathogenic avian influenza, chickens, and mealworms all go together with K-State Associate Professor of Veterinary Virology, Laura Miller. Laura, thanks for joining us today.
Oh, thank you for inviting me.
So as we're going to talk about HPAI, chickens, and mealworms, before we talk about how they're all connected, can you remind us, what is HPAI?
So HPAI is highly pathogenic avian influenza, and it's a global issue. There is low pathogenic avian influenza that is in wild birds and in poultry, but it really doesn't have that clinical severe disease. It's more respiratory disease. But when there's a mutation in the H5 or H7, which is the hemagglutinin, which is the antigen, one of the antigens of influenza, it changes the virus to be more virulent and more severe disease. So actually killing poultry potentially very quickly. It's sort of hemorrhagic and it rises. And so there's a systemic disease epidemics and a high mortality, sudden death of chickens. So, and the concern also with high path avian influenza recently is that jump it makes into other species. So recently into the mammalian, into cattle last year.
Do we see a tie to this? We talked a little bit about not just Kansas, but is there a tie to a first-time experience in Kansas with HPAI?
So just recently there was an outbreak in Kansas with HPAI, and so it was one of the largest outbreaks in this current epidemic, which is the U.S. So since 2022 to 2026 in the U.S., 131 million layer chickens have been depopulated because of various outbreaks across the states. And so the other in Kansas, this is sort of shown these outbreaks, but the other concern is that it's constantly mutating. It's a fast-moving virus as it spreads. And so, as I said, it jumps into mammalian species is one of the concerns, or into humans, but also that it constantly changes. So you won't even get that immunity naturally to the disease.
At the beginning, I mentioned HPAI, chickens, and mealworms. And so how do mealworms tie into all of this?
So the mealworms, one, they're a supplemental food for birds. So it's a natural feed, and they're kind of fatty, and they produce proteins as well. So quite nutritious. And with this concept, we're working with the Insect Farming Initiative, who make the mealworms. And so the idea was we can gene edit these mealworms. And in fact, in our studies, we have another model, a red flower beetle, which is similar, but we know more about that genome than we do the mealworm. It's got life cycle is a lot faster, so we can do things with the genome and see results faster than with the mealworm. But that's so we're doing the two models. So the mealworm kind of follows slower, but that's where the advantage of can we express the antigen to influenza in these mealworm that it produces the protein and then has an immune response within the chickens. So that's the concept of having that immune response with basically an antibody response, like to a vaccine, to the antigen that these mealworms are producing. And then the nice thing with the mealworms as a concept is that it's just feed. We're not having to inject chickens, so we're not having to do all the handling, stress for the chickens, using injections, all the people coming through, so then again breaking biosecurity because you've got more people, and trying to do that, vaccinate 1 million chickens quickly is not going to be easy with workers. So if we can do it through these insects, that's going to work much better with the larva, the mealworm larva. Also, it's cheap to produce because as the life cycle and as they grow, they're fed on garbage, basically, composting almost. So cheap to feed, cheap to grow, faster to grow than other, like if we were making vaccines in cell lines or eggs. So there's a lot of potential here. Also, we don't really need a biomanufacturing facility just in one place. We can have insect farms near to poultry farms to produce this. And then the other concept here is we can make one antigen, but we can also plug and play. So we can switch that as the virus evolves. Say we are seeing new outbreaks in our surveillance, we can quickly change that to whatever is coming and make those vaccines, 3 or 4 months.
For the mealworms, do they have to be alive, dead? How does that work for making sure the chickens would per se get that vaccine?
So the chickens like the live ones, they'll peck away in there, but they also can be fed freeze-dried. So we are doing the concept looking at the live ones, but as a safety aspect, if needed, we think this would be safe as it is, but if needed, we could feed them freeze-dried. The nice thing with the mealworms is also they'll just propagate themselves. So once you've done this altered genome, we can just grow it up. So we kind of want the live ones to grow in our farm, but then we can either freeze dry them and feed them that way.
So if they are like reproducing themselves, do their offspring have that same gene editing, and does it also mutate?
So the gene, because of the way we don't have it's gene edited, we hopefully don't have off-target effects at all. So then they would carry that, and it would just stay the same because of the region that we're selecting and the way we've put in all the little other things we're adding to. So just sort of in the sequence to see how it's expressed, developing that. Also, we adding some targeting molecules so that it goes towards the immune cells in the chicken gut. So it's reaching the cells that we wanted, so we get a better immune response that way. Also, some of the genome helps that we're targeting in the genome that it is where protein expression happens. So we have an increased protein expression, so we get as much as we can for that. So there's lots of elements to the concept that are a little bit fiddly, but we can optimize and perfect it that way.
With optimizing, hopefully this mealworms and helping with HPAI and poultry, is there any concern with them having too many mealworms if these mealworms have been gene edited?
So one of the things we will be investigating is the dose. We kind of know what dose of antigen we need from other studies with potential vaccine, but getting that protein expression and knowing how many mealworms we want to do. Potentially there wouldn't be you can overdose necessarily on this. Now we do have the question, will they become more tolerant the more exposure? But hopefully this would be just a dose and a boost like any other vaccine treatment that we would do. These are the studies we do in the second and third year. So we make these gene edited, and then the second year we're doing immunogenicity studies with chickens. So we have a layer population that's our sort of sampling species that we want to use of layer chickens. We'll look at that immune response. So we're doing a lot of assays looking at the hemagglutinin inhibition, virus neutralization, T cell responses. So a lot of the immunology side, that's with Dr. DeCal in his lab in our same department. And then we will also, the third year, we do an efficacy study in the Biosecurity Research Institute. So that will be with Dr. DeCal's group and my own group, Dr. Schertzinger. Schertzinger in my group is the one that's making the construct and some of this concept idea of it. So in the BRI, we will be doing efficacy studies. So that we will vaccinate chickens and then test with virus and see if it prevents transmission of virus, how good immune response we get to that. So some of those studies to establish, does this work against what we're looking for it to work against? And are there any other things that we need to see? So the first study, immunogenicity, should answer the safety questions and are we getting immune response? The second should answer, does this work truly in the field?
as you guys continue to do all of that research, will there also be an aspect of making sure that the consumption of those chickens is still something that's safe?
Yes, so the nice thing with this cooperative agreement with APHIS funding it, so USDA Animal Health Inspection, is that it is a work with them, so regulatory bodies. We have a program manager that works with us, and we have even members of the FDA. Now, the chicken should be safe because it is an immune response. There's no editing to the chicken happening, only the insects, and all the insects is producing is a protein. So theoretically, it should be very safe, but we also have these members, and we have support also from industry that already produce vaccines that also can tell us how the scaling up of this into a product, the regulatory issues that may come up. So we're learning with a lot of buy-in from other people, so it's very helpful.
That was Kansas State University Associate Professor of Veterinary Virology, Laura Miller, as she discussed how highly pathogenic avian influenza, poultry, and mealworms all go together. We're cutting to a short break on agriculture today, but when we come back, we will be joined by Laura as we continue our conversation on HPAI, poultry, and mealworms. If you are reaching your destination and aren't going to be able to hear the rest of today's show and would like to hear more on this topic, you can find the rest of today's show by going to agtoday.net or by searching for Agriculture Today on your favorite podcast platform. If you'd like to read more on this topic, I will link an article titled Protecting the Nation's Poultry Supply in today's show notes on agtoday.net. We're cutting to that short break now, but when we come back, we'll once again be joined by Laura. You're tuned back into Agriculture Today, and we continue our show and conversation with K-State Associate Professor of Veterinary Virology, Laura Miller, as she continues discussing highly pathogenic avian influenza, chickens, and mealworms. Laura, we were mostly talking about poultry and chickens. However, do you feel like there was a tie-in when it comes to wildlife as well?
Yeah, so the ideal is a nice broad protective across different species. Now, we may have to do experiments to see if this works in turkeys or in other especially the dose and things like that, and whether they like mealworms. Now, potentially there's other insects and other insect larvae we could use that they like better. The concept should still work, but potentially wild species, because it is an oral vaccine, is it something that we can put out there that could be used for wild birds? There's probably a lot more information to gather on that, but it is a possibility if we, it's in the same concept as rabies vaccine is given in Europe, that kind of idea.
Do you see kind of this concept of mealworms and chickens and HPAI being used for a different species, a similar modeling?
Yes, I think potentially as this mealworm farming grows and being used for other species, using it as a protein source for feed already. It is with insects and even in pet food or in, there's potentially, because it's just a protein source, it could be used for many other viruses that could, if they potentially have a vaccine of this concept, doing antigen, it should work for something else. So, if framed right, yes.
And Laura, we obviously have a focus towards Kansas, the United States, but do you see this also being something that could be potentially applied globally?
Yeah, so HPAI is a global problem. This outbreak now in Europe, they kind of live with these epidemics constantly happening in small outbreaks. They do vaccinate in some places in Europe. But also in developing countries, it's a concern. In the Indo-Pacific areas, places like that, where poultry is their main protein source. So it's very important for them to have some kind of treatment or vaccine or be able to have that biosecurity. Also for America, even to have that biosecurity for transboundary disease. So moving through particularly these highly pathogenic ones as well. So the potential with this being insect feed is it's a lot easier to give. So maybe even the backyard farmer could use it. And so farmers in developing countries could easily use it. Using the insects means we don't need that cold chain for a normal vaccine. We don't need to refrigerate it. We're just moving insects, growing them up at an insect farm near where we need it. So there's a lot of potential of better distribution methods, vaccine delivery methods globally.
Who and what has made this research possible?
So this concept actually with Dr. Schertzinger here in my lab, Dr. Lawrenson at North Carolina State, and Dr. Oppert in entomology was already conceived prior, but the right grant proposal came along with APHIS. There's $100 million that the Secretary of Agriculture gave to innovation on the concepts of vaccines, biosecurity, and diagnostics for HPAI has really given us the chance to look forward and really go after this and then for me to come forward and put these collaborations together and put the concept all together, get all the right people in, see if we can work with VRI, entomology, diagnostic medicine here. Even the veterinary diagnostic lab here in K-State is helping us because they have some of those recent Kansas samples. So we know that we're pinpointing that recent Kansas outbreak. And also, we have our poultry pathology experts there with the veterinary diagnostic lab that can help us with some of these items that we see with the poultry when we're doing that. We have the immunologists, we have entomologists. So it was the right timing with what APHIS are looking for, and it was the right group of people getting together in a like-minded concept, a big idea, and it just came together right.
So, Laura, as you continue working on this for the next few years, what makes you excited?
Well, I'm excited about, I always like educating graduate students and getting these new techniques to them. So we have graduate students involved in this and even undergraduate students involved in this study over in entomology and with our lab. So actually they're getting that interdisciplinary or transdisciplinary expertise for these students. So that's really exciting for me, the mentorship. Seeing that people putting these teams together is a lot of fun. And then I have a lot of other collaborations going on into further veterinary vaccines, trying to get these vaccine delivery methods for global. So we have interactions with other countries and collaborations in other countries potentially for many different diseases. So my concepts are many disease agnostic. So different species of animals and different viruses that I work on, and even some bacterial complexes with viruses that I'm studying as well. So there's a lot of opportunities I can see with this, but it also complements my other studies. I'm very much looking at the whole story. So the systems of immunology, how the response to the vaccine happens, and how we can improve on vaccines from what we learn. So it's kind of a circular information that comes back as well. So that's kind of what I'm really looking forward to, is making these teams work and seeing success and keeping things on track and these interactions. And even that one of the other pluses here is that collaboration with industry. So the entrepreneurship and biomanufacturing side of things that's new in this field.
If people are wanting to find out more information about this or kind of keep up to date as you're working on it, is there a way they can do that?
So we do quarterly reports with APHIS, and I think they publish them as they go through the USDA system. That information will come out because it's kind of a well-managed program management. And there's a number of other vaccine studies. So 17 studies were funded. In fact, at K-State, we got 2. So myself and then Dr. Mwangi's lab next door within the same department. So this was a big call, this grand challenge call. So the $100 million towards this kind of research and highly innovative studies. And then 58 of those were funded, 17 for vaccines, and then others for diagnosis and detection. So there's also not just universities, but other industries. So when I spoke 2 weeks ago now at the USDA Agricultural Outlook, they had also speakers, one with detection, so developing PCR, and that was from Canon. And then another group, Radio Life, looking at other detection methods for virus for surveillance. So kind of interesting different groups and interacting with them as well, learning new technologies. So there's a lot coming out that's really exciting.
Anything else you'd like to share, Laura?
I think that we wouldn't be able to do this if it wasn't for K-State, because of what we have put together. We have the strong USDA presence, we have the strong College of Agriculture, we have the Veterinary Diagnostic Lab, we have the Biosecurity Research Institute, we have the Entomology and Insect Farming Initiative. So we're very fortunate. And then we have our good collaborators at North Carolina State. They're helping us with that genetics and gene editing.
Laura, I appreciate you taking the time to join us today.
You're welcome.
That was Kansas State University Associate Professor of Veterinary Virology, Laura Miller. As I had previously mentioned, I will link an article that was done on this topic in today's show notes on agtoday.net. As always, if you're wanting to find more information from K-State Extension or Kansas State University, you can do so by going to Extension.ksu.edu or by going to K-State.edu. Before we cut to a break, we're now going to be joined by USDA's Rod Bain and Brian Combs of the National Agricultural Statistics Service as they discuss what some of the findings are within USDA's most recent Total Ownership and Transition of Agricultural Land survey.
How many acres of U.S. farmland is rented to ag producers for growing crops? According to USDA's latest tenure, ownership, and transition of agricultural land survey,
those landlords collectively rented out 350 million acres of farmland.
Brian Combs of the National Agricultural Statistics Service says acreage for 2024 applied to farmland owners who are both operating farmers and non-operating landlords.
There were over 2 million landlords who leased agricultural land to farmers. This is a slight increase from 2014.
The last time this once-in-a-decade survey was conducted. Additional findings from the total survey, rented farmland acreage, including buildings, are valued at $1.6 trillion. Landlords in 2024 received over $34 billion in rental income, while incurring $12 billion in operating expenses. I'm Rod Bain reporting for the U.S. Department of Agriculture in Washington, D.C.
Once again, that was USDA's Rod Bain with Brian Combs of the National Agricultural Statistics Service as they discuss the Total Survey, which stands for Tenure, Ownership, and Transition of Agricultural Land. Before I cut to a break, I would like to let you know about an upcoming update from K-State's Antonia Boryaka that she'll have on March 25th. This webinar is titled Ukraine-Russia Conflict, Agricultural Ramifications. As always, there is no fee, but registration is required. You can find it on agmanager.info. We're cutting to a short break now, but we'll be right back.
Welcome back to Agriculture Today. Along with Shelby Varner, I'm Jeff Wickman. We end today's program with this week's milk lines. K-State dairy specialist Mike Brouk says an increase in herd numbers helped produce a banner year for the Kansas dairy industry in 2025.
Today I'd like to talk to our Kansas dairy producers as well as the general public about 2025 and what that has meant for Kansas in terms of our dairy industry. 2025 was a very successful year for the Kansas dairy industry in terms of increasing cow numbers as well as overall milk production. We added about 45,000 dairy cows to our dairy herd in 2025, and that resulted in a large increase in milk production. Our year-over-year milk production increased by 17.2%, and that actually led all states. Second place would have been South Dakota with a 9.5% increase. Overall in the U.S., milk production did increase in 2025 over 2024, about 2.6%. So we saw about 5.8 billion pounds of additional milk production in 2025 over 2024. And 2025 was an increase that we had not realized in 2023 or 2024. So banner year for the dairy industry across the United States as well as here in Kansas. As we look at what's happened over the last 5 years, Kansas also has seen great increase over the last 5 years. about 22.9%, and that puts us in second place, just behind South Dakota, that saw an increase of 73.8%. As we look at states next door to us and what's happened to milk production there, Missouri in the last 5 years has realized almost a 31% decrease in total milk production. New Mexico's down about 28% in the last 5 years, and Tennessee down about 25% as well. Even Nebraska to our north is down about 15.4% for the last 4 years. So as we look at where we rank as far as a state in total milk production, in 2025 we rank 15th, which is a nice increase. And as we look at per capita milk production in 2025, so that's a comparison of what milk we produce in the state divided by our total population. And we sit at 1,663 pounds per person, which is a net exporting state. So we export a lot of milk out of Kansas into the southeastern United States. And those states like Missouri that are experiencing production decreases, as well as in the deep southeast, we provide raw milk to be utilized in the processing plants down there to balance those plants out and provide for the consumers in the southeastern part of the United States. So what does this all mean for production here in Kansas? Well, the numbers for 2025 are not fully reflective of what our dairy herd is actually capable of. Many of those animals were added in the last half of 2025, so the total production increase for the year is only based on about 6 months of increased production. So moving into 2026, I would look forward to Kansas seeing a fairly sizable increase in total milk production as we get to a full year out of those additional cows. I also expect that we will see some smaller increases in dairy cow numbers in the state, not expecting that 2026 will result in an increase in another 45,000 head, but I do think there will be a few thousand head added to our total inventory, so this will also increase our production. As we get closer to filling the plant in Dodge City, the Hillmar plant, we will also see some increases in cow numbers to do that as well. So exciting times in 2025 in terms of production increases for our dairy industry here in Kansas. We look forward to a bright future as we move into 2026. This is Mike Brouk with K-State Research and Extension.
Thanks, Mike. And that'll do it for this edition of Agriculture Today. For Shelby Varner, I'm Jeff Wickman. This is the K-State Radio Network.